everybody and welcome to the ISI Sementi Field Days 2021. My name is Anna Giulia Boni and I am the head of the Laboratories. So today I wanted to give you a brief explanation of what we're actually doing here in our labs. So first things first, we have three laboratories. The first one is the Quality Control Department, then we have the Phytopathology Lab and the Molecular Biology Lab. All these three laboratories work in two directions. The first one is the quality control of our products, and the second one is the research. So let's dig a little bit into these two aspects. The first one is the quality control, as I said. So all the seed lots that came in from our production site of all the crops are tested for different parameters. For example, humidity, physical purity, vigor and germination by our quality control department. Then they are tested for genetical purity by our molecular biology lab, and last they are tested for the seed health by our phytopathology department, so different pathogens for different crop, of course. Then, as I said before, there is the research. So all laboratories help the breeders to create the hybrids, so the hybrids that the market and so the clients need and ask for. So different labs do, of course, different things to achieve this goal. For example, the quality control department assures the breeder of the vigor and germination of their lines for all the crops. Then we have the phytopathological department, that is our newbie, is our newest lab, and they test different crops for different pathogens. So they infect the pathogens in order to identify the resistant plant or the susceptible plants. So the breeders can actually carry on just the resistant plants. So we have internal protocols that has been developed here, internally of course, for different crops, such as tomato, pepper, onion, lettuce and many more. And last, we have our molecular biology lab. In our labs, our technicians work with molecular markers. So these are the short pieces of DNA that are strictly linked to a gene of interest or within a gene of interest. And for gene of interest, I mean genes that code for trait of agronomical interest, such as fruit shape or color, or the resistance to some pathogens. So our technicians extract the DNA from a single plant, and they can test the single plant for more than one molecular marker at the same time. So the breeder can actually select a single plant from more than one trait at the same time. And so this process helps the breeder to really speed up its activity in the fields. Okay, so this is more or less what we actually do in our laboratories. I try to keep it simple, so I hope everything is clear, and enjoy the rest of the tour. Bye!